here's what you need to know about the cell. So, here are all the internal components. So this is just a singular cell. So, if we just kind of take this piece by piece, so all of these things that you're seeing here are what are called our organelles. And so the first one that we're going to start with is the one that's going to be on this outer side here. So anything that's on the very outer ring is going to be called the plasma membrane. So this is actually going to have different proteins in it. This is what's going to protect the cell. So essentially the plasma membrane is going to act like the door to the cell. So without the plasma membrane, things could just go in and out freely. So the plasma membrane actually acts as a barrier. So it allows some things to pass through and some things to stay in. So this is one way it's able to keep the internal and external environment separate from each other. So going further from the plasma membrane, the white portion here that you see that's all around this would be referred to as the cytosol. So this is essentially what all these organelles are floating in. So please remember, this is just a model, but in our cells, they're actually floating. So think about the consistency of Jell-O, and essentially that's what the inside of our cells are like. It's kind of funny to think about that our cells are kind of made out of this gelatinous mixture of Jell-O, but that's kind of the best consistency that we can really give. So all of these organelles are actually floating in it. So one that you might be familiar with is the mitochondria, which we all know is the powerhouse of the cell. This is responsible for the citric acid cycle, as well as oxidative phosphorylation. So this is what's really producing a lot of our ATP, or adenosine triphosphate. Now, you see that there's abundance of these because we need a lot of energy. So there's usually going to be, some cells have a lot of mitochondria versus other cells, but especially like our muscle cells that need a lot of energy to move. So another one that you might recognize is the rough ER. So one reason it's called the rough ER, if you see these little red pieces here, you might can hear it. So it's actually rough. It's called rough because there's actually ribosomes that are attached to it, and it gives the appearance of being rough. So along with rough ER, or rough endoplasmic reticulum, you have a smooth endoplasmic reticulum, and it actually does look the way that it sounds. It is smooth. So the rough ER has ribosomes on it, which ribosomes are for protein synthesis. So things like insulin or the proteins that make our skin, that comes from the rough ER. As for the smooth ER, that's going to be dealing with lipid synthesis, which lipids are fats essentially, and that's going to be dealing with the smooth ER. The next one is the Golgi apparatus. The Golgi apparatus is also known as the mail center of the cell because it tends to package things and ship it outside the cell. So that's why you see these little bitty vesicles going towards the plasma membrane. And remember, the plasma membrane keeps things in and out of the cell. So therefore, it packages them and sends it outside the cell. Another thing you might notice here is the centrioles, or the centrosomes, which tend to be parallel or perpendicular to the nucleus. So the centrioles are responsible for making microtubules. They're usually used for cell division when it comes to splitting those chromosomes away in mitosis and meiosis. So that's usually the function of those. This here on the very center, this whole thing is referred to as the nucleus. However, there's different parts. So this very outside layer, if you think about a letter that you mail, this would be referred to as the nuclear envelope. And these little bitty holes inside of here are called the nuclear pores. And this is where RNA will actually come in and go out of the nucleus. Because remember, the DNA, or this chromatin, which is this red piece, this little really straggly piece, is really just DNA that's free floating, also known as chromatin. The little gray portions in here are referred to as the nucleolus, which are going to be for protein synthesis or ribosome synthesis. That is what the function of the nucleolus is. Another organelle are these two here. So you have one that is lighter, and then you got one that's darker and a little bit bigger. So the one that's lighter is going to be the peroxisomes, and it kind of sounds like peroxide, and that's actually how it got its name. So this actually helps break down. These are essentially like the garbage disposal of our cells. So these actually take old proteins or misshapen proteins and actually breaks them down for either to be recycled or to be shipped outside the cell to be destroyed. Lysosomes kind of do the same thing. They actually have enzymes that are going to break down proteins as well. So they actually kind of work together. They both use enzymes. Now, a place where you might actually find these, you'll find lysosomes, especially around in our eye. So when you are crying, if you get dirt in your eye and your eye starts to water, there's a bunch of lysosomes in your tears and around your conjunctiva of your eye that are going to actually break down those proteins or the bacteria that get into your eye. Peroxisomes, a place where you can see those would be if you eat an apple and you see that it turns brown after you leave it out because it starts oxidizing. That is due to peroxisomes actually are starting to break down the area where you have bitten. So that's a way you can actually see some of these things. All right, so that wraps up the cell. Hopefully you learned something new, and we'll see you next time in our lecture series.